Welcome to Sigest, your fortnightly serving of digestible science from plant and food research.
Hello and welcome back to Sigest. My name's Blue Plunkett and I'm one of the Sigest hosts. Today we're here exactly 5 years since our first podcast and over 100 episodes later, and we have today Dr. Andrew Kralicek. Hi Andrew, thanks for coming out today.
Hey Blue, it's great to be back. I can't believe it's been 5 years. It feels like a century.
Yeah, it's amazing to have you back because you were also the first podcast as well. So it's great to have this update from you today. So on the first podcast 5 years ago, we sort of went over some background, and at the time you were a scientist at Plant and Food Research, but since then this company you've been building has really taken off. So I was wondering if we could just go back to 2017 and go through a bit of that process.
Yeah, I was leading the molecular sensing team here at Plant and Food Research, and we had been doing about 15 years research on insect odorant receptors and how they functioned, and with an interest to see whether we could use them for a purpose, so in an applied way, because there's huge potential for what they're doing. And so at that point we were just starting on our journey of commercialization, and we'd just secured some IP, some data for IP, so we got some patents, and that was the key step to get us going in the commercialization journey, because that's something of value to take to investors. And I guess I thought at that time it was going to be really easy. We'd go, in a year we would have the company started, but it took much longer, because not only did we have to get the IP patents submitted, but we also had to build the business case for it, to understand where the value was. And you mentioned that we were looking at biosecurity, which is a great need, but in the world of investing, investors are looking for a return on investment. And biosecurity is not something which is going to give a big return to investors. So we were looking at other markets. So we looked at wine quality initially. We looked at then food safety as another potential low-hanging fruit to get a win with our technology. But in the end, we settled on flavour and food quality, which is a big market and where there's a really big need. So there was the IP, there was the market, and the other thing was to be able to demonstrate a prototype, to be able to prototype our device. And that was a journey in itself too, because we first started, okay, let's go build our own device. You know, we've proven these receptors in the lab on a variety of sensor technologies. Let's go make one. And that proved to be very challenging. We worked with electrochemical impedance spectroscopy, but unfortunately it wasn't robust enough. So we had to then look for another direction. And so we had a really cool interaction with a company out of Oxford University. Fantastic technology. They came down to New Zealand for 2 weeks in 2018 and we got it to work. And we were amped, right? We've got it. Well, let's do a joint venture, between PFR and this company. But 3 months later, they were gone because they were a startup and their investor pulled money on them. And so that left us in a real hole. What do we do next? But fortunately, we found a company out of California who had another platform which we had IP on, did a pilot study, and it worked. And so we had the business case.
Nice. So yeah, these three key things sound like such a thing to chase all at the same time. So for the IP, it must be different for each country. So you have to do a different sort of application at different markets, I guess.
Yeah, it's you've got to write the thing, which is an exercise in itself. You have to have the data which can prove the claims you want to make, and it has to be original. And basically, we've applied for it in various jurisdictions. And I'm happy to say that we've had our first patent. We've got 2 patents. Our first patent's accepted in China, Taiwan, Japan and Europe. So we're on a good run.
Nice. And then so once you've been able to protect that, is that what allows you to work with these potential companies overseas, or you had sort of had shared IP, or was you that had the IP and them that had the platforms? Just wondering how that's structured. So
the way it's structured is that Plant and Food Research owns the IP, and they've licensed it to Scentian, the company I've founded. And what that means is that we can approach sensor technology providers and say, we've got IP on your platform. Would you be interested in working with us to develop a product?
Gotcha. And so it sounds like it's quite a big step to go from, okay, we've got this insect receptor detecting what we want to in the lab, and then sort of getting that to a prototype that you must have to meet thresholds of reliability and scaling and things like that. So has that been quite a big part of the challenge?
That's been a major part of the challenge, and it's an ongoing process. It's all about robustness, or reliability, as you're saying, and the ability to scale, to mass fabricate sensors. And indeed, this is probably one of the biggest challenges for biosensing in the world right now. I was quite surprised how early it is still in the real world to do this kind of thing. And you can imagine it, Blue, when you think about the biosensors that are out there, which we encounter in everyday life, there ain't much. There's pregnancy tests, there's the COVID test, the rat test, and there's probably sugar indicators, for diabetics to test, and that's an enzymic reaction. So there's nothing else, right?
So how long has the science been around?
Yeah, well, science has
how's the field?
It's been around for about 30 years, right? There's tons of patents and papers out there, but there's a challenge to go from the lab out into the real world, right? It's called the Valley of Death in general for startups, but there's this real challenge to be able to make that jump. And there are some companies doing it, which is fantastic, and it will come, but it's not as developed as I had thought when I started this journey. That's been a big learning for me, yeah.
And so these companies that provide platforms, as opposed to trying to come up with your own platform, what's the sort of advantage there? Is it a sort of a mutual collaboration that benefits both, or it's just too difficult to be a platform company and a receptor development company?
Yeah, I think we're kind of in an interesting position. Most of these biosensor companies have a platform which they want to commercialize, and they will say, we'll do antibodies or aptamers, these DNA sensors, and that's the usual go-to path. But taking it out to other kinds of receptors, no one's really done that yet, right? And for Scentian Bio, our company, we're about the receptors. We know how to make them, we know how to handle them, we know how to couple them to sensor technologies, and we're about the data. We know how to interpret the data and how to pick the right receptors for the right application. But what we're not about is the biosensing. That's not our forte, and we don't want to reinvent the wheel because there's plenty of people out there who are doing fantastic work on that. So we're looking for partners who can provide the platform that we need for a particular application. And indeed, it may be for one application, health diagnostics is one platform, or for air quality sensing, which is another application, it could be a different platform, right? So is the word agnostic probably the right word in terms of what platform we want to use for particular purposes? And I think that's to our advantage because we can't do everything. Startups are small. Our startup, there's only me and one other person who's gone out with the company, and then we've got 3.5 other people more or less who are on a service contract from PFR who we're hoping to bring out. But, we're not Plant and Food Research with 800 employees. So, yeah.
So before we go too far into the details, I was just wondering what exactly is a biosensor and what does it detect?
Okay, a biosensor is a take on a sensor. So a sensor is usually an electronic device which can sense something. It could be a compound, it could be a protein, it could be light, it could be sound. So a biosensor is actually harnessing nature. It's taking biological molecules and putting them on the sensor and using their power to detect things. So in our case, at Scentian Bio, we're taking insect olfactory receptors. They've been evolved over 400 million years to detect volatile organic chemicals. I think they're probably the most unique proteins in the world because there's nothing like them. When you think of the sense of vision, right? It's amplitude and frequency of light. And we've evolved 3 receptors to detect that. How the hell do you detect inordinate complexity of the chemical world, right? And what nature's done is to build these families of proteins, but they just don't have one protein detect one compound. One protein can detect hundreds of compounds, and the way it responds depends on the shape of those compounds. So a biosensor, just to reiterate, is about using a biological molecule to detect something electronically, usually, yeah.
So yeah, that's amazing to be able to harness what nature's already come up with in far as these receptors. And so the molecule gets bound, and then how have you managed to stabilize the insect receptor so that you can send it from this sort of biological protein to a digital signal?
Yeah, so that's a really good question, and it's not easy because these are proteins, but they're membrane proteins, so they're probably the most unfriendliest proteins you can have because they hate water. They live in lipid membranes, like fatty membranes. And so you have to be careful with them because if you take them out of those membranes, you can knock them about and they won't function. So what we've worked out is how to put them in display forms, and one of them's in liposomes, which is literally a vesicle of water with a bilayer on its outside, and they're sitting in there. We've put them in things called nanodiscs, which is basically a protein inserted in a disc of membrane. It has a belt of protein around it to keep it stable. So when you can get them into these forms, then you can treat them like normal proteins, which are soluble in water, and you can work with them, right?
Well, this idea of nanodiscs sounds amazing. So is it essentially you've sort of made it at home like it's in a layer of lipid where it's normally found, and so it stays stable and functional? Yeah,
exactly. I don't kill insects. I'm not a serial killer of insects. We basically make the genes. We take the genes of these insect receptors. We cause their production in insect cell lines. We grow them up. We break them open. We extract them from the membrane using detergent, like soapy water, and then we put them into these different lipid, these ways of presenting them in membranes. Yeah.
Very cool technology. So once you have it safely on the nanodisc and it detects the molecules that it can detect, is it some sort of change or charge difference, or what's the technical effect that it has on the protein?
You've got it in one, and it's still not well understood because we don't actually have a structure of the insect olfactory receptor complex. We have structures of the components of it, but it's down to a structural change when it binds the chemical. It's like a hand grabbing a ball, you grasp it. And that change causes changes in the charges on the surface, which can then affect the flow of electrons through a sensor surface, right? So that's how you see a signal change of binding.
Amazing. And so when, just in my life passively hearing about insects, they're very sensitive to low levels. Are you able to achieve the same sort of sensitivity as insects?
Yeah, totally. The great thing about insect olfactory receptors is that we've shown that when we put them on sensor surfaces, they're 1,000 times more sensitive than the dog's nose, right? Which is, a dog's nose can go down to parts per trillion. We've gone down to parts per quadrillion. And just as a measure for us to smell, it's parts per billion. Yeah. And so they're amazing proteins once you take them out and put them on sensor surfaces. And in comparison with technologies out there right now, the gold standard is GCMS, which can get down to parts per trillion, normally in parts per billion range.
So you're an order of magnitude or two above sort of current standards.
Totally. That's where the hope is, that we can take these receptors, we can miniaturize them, we can automate them and put them on chips. And then you don't need these massive machines in a lab and a guy with a PhD operating it and trying to interpret it. It's something that everyone can use, right?
Does it need to be reset after use, or is it sort of something that's changed out each time, or can you just redetect with the same sensor? So
that's a really good question, Boo. Those options are on the table. So there's different ways to think about using these receptors. It can be like a one-off shot, like you
took this shot,
and then the photograph would come out.
Oh, the Polaroid,
yeah. So it could be like that, or it could be like a video camera, where you're constantly measuring. And in some ways, we see this technology as a camera for smell and taste, because it's a digital record of the aroma which is present in the air or in the liquid that you're tasting.
That's a cool way to think about it, a video camera for smell and taste. What would be the sort of target use case, or what's the first volatiles you're after to detect?
So that's another good question. The thing with this technology is there's so many opportunities, right, in all types of market verticals or application spaces. And I mean, health diagnostics is a big one, this idea of being able to non-invasively detect disease, or your state of wellness simply by breathing on a chip, or taking your urine, or maybe blood, for example, or saliva, because they all contain volatiles.
And so certain diseases would be associated with particular volatiles on the breath?
Exactly. There would be a fingerprint associated, because really when you think of your breath, or your lungs, it's the window to your state of health. Like your eyes are, they say it's your window to your soul, right? Because whatever's happening in the body, it's generating these metabolites, which are in your blood, and they go to the lungs, and they're immediately released out as there's air exchange, oxygen exchange, and CO2 exchange. So they're on your breath. There's over 2,000 compounds on your breath. And I like to call it a chemical language, which is waiting to be interpreted, right? Because it's all information about the state of your body. Yeah.
And so you mentioned currently needing someone with a PhD with a big machine in a lab. What sort of size and mobility and things would your detector be?
We're currently looking at doing a prototype which is probably a bit bigger. Well, the chip's probably roughly the size of a smartphone, or a little bit less, with the reader included. But the end goal would be to get it down to something which could be incorporated just like your camera is in your phone, and have that there. So that's just another tool which is available for you to interact with the world, right?
It is amazing watching phones absorb more and more appliances and things.
Yeah.
I still have to carry my wallet and my keys, so I'm looking for those to get into my phone too. But
I don't think you need them anymore, man.
Well, it's probably already there. True.
That's right.
But yeah, it'll be amazing to have something that's detecting volatiles on your phone as well for medical diagnostics. That's amazing. And so that sort of brings us closer to today. Where are you sort of at the moment? You've had some good news recently regarding some investment.
Yeah, it's been, maybe I should just pick up from where I left off, which was where we'd done the raise, we'd got this platform, and we spent, about a year. Well, I had a 14-month runway, and then, of course, COVID hit, and that stole about 6 months of that, which really raised the pressure. But unfortunately, it didn't work out, okay? We couldn't get that platform to be robust enough to do what we wanted to do. So we had to walk away from it. And that was at the start of this year. And basically, we've been on this searching the world for the platforms that are out there and whether our receptors would work on it. And I had a member of my team, Colin Cutter here, who was sent out like, I don't know, SAS soldier, went to the States, went to France, went to the UK, visited these companies, engaged with them, did some test runs with them, and had some successes. And all that information we had to digest, and basically we came up with a new way forward, which is a really exciting way forward. And the good news, which I can't tell you about because it's still kind of embargoed, is that we've got now a significant cash investment to go in the direction with this new approach. I can say it's in the health diagnostic space, so which is really exciting because it just seems the right way to go. Yeah.
Yeah, congratulations. That's awesome. As far as other companies or competition, is there much other companies that you're sort of in a race with?
Yeah, it's like the gun's gone off, right? And in startup world, probably started about 10 years ago. Some companies started off trying to use human odorant receptors and they failed, they died. There's a company called Aromix, which has been around for probably the last 10 years, which is still going, which is great to see. And it's using human olfactory receptors, which are different to insect receptors because human receptors are G protein coupled receptors and insect receptors are ion channels.
Is that a difference in just how they operate or their structure or just across the board they're quite different?
Across the board they're different, but insect odorant receptors, when they're turned on, they cause a flow of ions through the plasma membrane, right? And you can detect that electronically. GPCRs are different. They require, I don't want to get too technical, but amplification of a signal from a G protein. And often when they're used in sensors, it's a structural change that you use.
Okay, so these human cells, it's a structural change, whereas for the insect protein, it's a charge
It's still a structural change, but it's an ion channel as the other bonus effect, really. So, yeah, Aromix has worked with cells and they've moved into cell membranes using a microtiter plate kind of lab-based instrument There's a company called Kaniku, which is doing really interesting work with living cells containing human receptors. And I think they've got a product coming out around sensing of potentially explosives, and they were doing COVID detection in planes potentially. There was another company, Yesi, which was doing something similar, but unfortunately they lasted 3 years and now they're gone, which is a real shame. And this is the churn of the startup world. And there's even now another insect sensor company called Insect Sense, which appears to be training bees to detect things. So they've trained bees to detect COVID, and that's interesting in itself, but they're building devices where you've got living bees in them. But for us, that's exciting because it just shows that we're going in the right direction if others people are joining the race. And I hope we're going to win, but I can't tell the future. But I do believe that somebody in the next 10 to 20 years is going to get a product out there which the everyday person can use for VOC sensing. And you won't even realise that it's doing VOC sensing, but it will be telling you stuff about your everyday life which you can't do now, right?
By VOC sensing, you mean volatile?
Sorry, volatile organic chemical sensing, yeah.
Gotcha, yeah. Interesting. It sounds like your approach is quite unique and differentiated insofar as some of the other options there, so that sounds really exciting.
Yeah, well, I guess it's because, we're humans, we're human-centric, and people are looking at human health or flavour and fragrance, and people have sort of not looked at insects very well, even though the insect nose or the antennae is probably the most characterised in the literature. So it's the most well understood. But I believe it offers the biggest potential because I think insects are more efficient at smelling and they're more sensitive. And of course, we've got IP on it, so I truly believe in it.
Yeah. And also the combination effects, how you say one protein can detect.
Yeah, so you've touched on something really which is fundamental to smell. It's a combinatorial response pattern. So a fly uses 45 receptors to detect thousands of different kinds of volatile organic chemicals. And that's like what I was saying previously, that because each receptor can bind many compounds and respond in subtle ways to those. So that's why we call it a fingerprint or a VOC print response. Humans use over 400 receptors. The dog uses 1,400 receptors. And so really, to get to a device which replicates an animal's sense of smell, insects, I believe, is the fastest way because you don't need so many receptors to smell. And one of the biggest challenges with this is being able to, you can make the receptors, but how do you prove they're active, right? So that's another whole talk.
Insofar as personal learnings, what sort of changed as you went through this compared to when you started to today?
Yeah, well, so my role is as CTO of Scentian. It's a Chief Technology Officer. It's kind of, I don't know if you know Catha Simpson, but she set up Engender Technologies and she founder of Orbis Technologies as well. And when I said that I was going to become the CTO, she told me, you're the cream between the cookies and an Oreo, right? And then Richard Newcomb, the chief scientist, was there too, chief scientist of Plant and Food, and he said, you're the piggy in the middle. And you're kind of a bit of both. When things are going well, you're the cream between the two biscuits. When things are tough, you're the piggy in the middle because you're really the pressure point between what the board is demanding, and they're demanding results, right? And you're the pressure point with the team underneath who are demanding the resourcing or whatever they need to achieve the milestones. So it's a different role to what I was used to. And the other big challenge, not only because of the demands of it, but it's kind of like you, as a scientist, you want to make decisions based on rigorous study. But in a startup, you don't have time for that. You almost have to make calls on your gut. And that's been challenging for me because I always want to know the facts before I make a decision, right?
Yeah, that's quite a change to have to adapt to.
Yeah, because you're moving at such speed. Yeah.
And so with this latest development where you can chase this new platform with the ion channels, what's the sort of next stage over the next 6 months?
Well, it's really about engaging with a tech partner, showing that we can get these VOC prints working on that platform, and then moving into a real-world study where we take something of value and show that we can get VOC prints and show that we can match match. So the way the technology is you take a VOC print with an array of receptors on a chip, and then you compare that with a library of VOC prints and see if you can get a match. And so when you get a match, you know what's going on with that sample, and you can probably also predict what the best action is to then do. And so that's our thing is next to demonstrate that on this platform. Yeah.
And so once you've been able to reliably detect these different prints, VOC prints from the library, then you've sort of proved your function, and then that's you're sort of ready at that point for
that's the next
for real life trials and things.
Well, it's the next, what they call in the investment, the next inflection point, because you've proven that's the next step. At the moment, we've got IP, we've got a platform. Doing what we're talking about, this VOC print stuff, is the next inflection point, because that's the next value point which you can raise more money on. And basically, startups are all about raising money. As soon as you've raised the latest batch of money, which this one will get us through hopefully to another 2 years, we got to start thinking about the next raise, and it's continuous. And so my role as CTO is I'm helping with the science, but I've got my team doing that as well. But I'm also out there talking to investors, talking to tech partners, talking to customers, promoting the technology and the potential of it. And it's a full-time job.
Yeah, the potential sounds great. Having something, in your phone that's detecting volatile compounds, I'd never even thought of that before. And just before we finish, I wonder if you had advice for anyone thinking of getting into this type of the startup area in New Zealand, if there's some sort of key points that you might recommend.
Yeah, I think it was a big learning curve for me. What I'd recommend anybody contemplating this is you need to do your homework, right? You need to get out there and talk to people. Talk to people who've founded companies, who've taken the step. Talk to potential investors. Talk to the potential customers where you think there could be potential for your technology to work. And I was really surprised and heartened to see that the investment community, particularly in New Zealand, they're very open to helping, and people will make time to give back. And that's great, because it's not the kind of decision you make every day to... In my case, I walked away from a permanent job. I couldn't get a parachute clause on this one, so I had to make that call. And there's risk in everything, and startups are risky, right? But if you don't take risk, you don't get the opportunity to do what we've done. And I know it probably sounds like a crazy journey as it is, a roller coaster ride, but we're still alive and we've got a great direction now. So that's the first thing. Do your homework, and don't trust things on face value. That's the other thing. That's also part of doing the homework. The other thing is really look after yourself. That's been probably my biggest learning is you got to take time out, because it can be very stressful, particularly when you've got milestones and time is running out and you've got boards saying, where's the results, right? So you've got to look after yourself. And so I've got a really good support network in terms of my wife and my kids and my family, who have been very supportive through some rocky times, and of course colleagues. And I guess two other things. Biggest lesson I've had to learn is to step up by stepping back, right? So letting other people do their thing, right? Because you can't do everything. And I guess previously I thought I could, but this beast we've got now, which is roaring along, I can't be everywhere. And so that's been a big learning thing for me, to step back. And it's also about letting other people grow as well. And finally, the other thing is realize you don't control the universe, right? You can only do your best, and increasingly I just think it's all about intention. What matters is intention. You can't control the results, but as long as you went in there with the best intention and made decisions on the best knowledge that you had at the time, you can't do anything else, right? So, and that's kind of a relief, actually.
Yeah, well, that's great advice, and seems like a perfect place to finish. So thank you so much for sharing all that, Andrew. It's an interesting story, and I can't wait to see what comes out of Scentian Bio next.
It's been a real pleasure, Blue, and I'll see you in 5 years.
That'll be great. I look forward to it. And thank you to our Sigest listeners for joining us on this episode. See you next time.